Good morning. Thank you all for coming today. My name is Stephen Perrin. I'm the President and Chief Scientific Officer at Eledon Pharmaceuticals. I'm also the original founder of the molecule that we used to call AT1501 and is now called tegoprubart. Eledon Pharmaceuticals is a publicly traded biotech company, and we're focusing on multiple indications targeting Cd40 Ligand, which is a receptor expressed on immune cells. And we have multiple phase 2 studies going in ALS and neurodegeneration, organ transplant, including kidney transplant, as well as cellular transplant with islet cell transplant for type 1 diabetes. And we're also in autoimmunity in an indication in the autoimmune nephritis is called IgA nephropathy. We have near-term milestones with all 4 trials reading out by the end of the year, with the first one reading out in ALS in late Q2. And IgA nephropathy, renal transplant, and islet cell transplant will be presenting data at the end of the year. We have a strong financial profile right now with over $85 million when we closed in Q4 of last year. So Cd40 Ligand and its receptor Cd40 are not new targets. They were actually discovered back in the 80s as receptors initially expressed on immune cells, with Cd40 Ligand being expressed on T cells and its cognate receptor Cd40 being expressed on B cells and other types of antigen-presenting cells. And what was interesting is when blocking antibodies were developed for the receptor versus the ligand and tested in animal models of disease, it became very apparent that when you block this signaling pathway, it literally can cure most autoimmune diseases. So it was tested in animal models of multiple sclerosis, rheumatoid arthritis, psoriasis, ALS, Alzheimer's disease, and others, and pretty much either ameliorates or stops disease progression. It plays a pivotal role as the second signal that amplifies an immune response, both when you get a foreign antigen, such as COVID-19, as well as when your body recognizes self-epitopes, like in the context of rheumatoid arthritis, where your immune system recognizes collagen as foreign and it attacks your joints. When you block this pathway, it blocks the immune system from recognizing those types of foreign antigens. Tegoprubart, formerly called AT1501, blocks this pathway by blocking Cd40 Ligand. We went after the ligand rather than its receptor because historically, going back to the 80s, blocking the ligand has always been much more potent than blocking the receptor. There's 3 high-level biological reasons for that, and one of them I've already mentioned. The ligand is expressed exclusively on the T cell, which means it's more restricted in where it's expressed in the body, as opposed to the receptor, which is pretty much ubiquitously expressed on all cell types that present antigens. So when you think about antibodies that would block this interaction, it's easier to think about an antibody specifically blocking the ligand because it's expressed in a more specific location. In addition to that, people often only think that the ligand interacts with the receptor Cd40, but in reality, Cd40 Ligand interacts with multiple different signaling receptors on many types of immune cells, and so it blocks multiple different receptors. And so when you block the ligand, you get multiple blocking mechanisms, more bang for your buck. And then the third one, which is probably most interesting, is because Cd40 Ligand is expressed on the T cell, not only does it block those pro-inflammatory cascades that I mentioned that cause autoimmune disease, it actually converts those CD4 cells into a special type of cell called the T regulatory cell, which is actually a cell that tamps down inflammation locally at the site of injury. So when you block the ligand, you get a very potent response compared to blocking the receptor. Tegoprubart's a full monoclonal antibody that we developed, and we went with an antibody because manufacturability of antibodies is well understood over the last 30 years. You tend to get very high yields. They have very good half-lives, which means you don't have to dose them very often. And finally, because monoclonal antibodies have very good drug-like properties, you tend not to have neutralizing antibody titers against them. And we've seen all of those as we've moved tegoprubart through clinical development. We completed phase 1 studies, single ascending dose studies in healthy volunteers back in 2019. If you look at the far right-hand side as a percentage, we saw almost no adverse events, and they were more common in the placebo group than they actually were in the entire AT1501 cohorts assembled combined. So a very good safety profile. We're focusing on drug development and neurodegeneration, with ALS being our primary neurological indication, with top-line data expected here at the end of Q2. By the end of the year, as I mentioned, we'll have data readouts in kidney transplant, islet cell transplant, as well as IgA nephropathy. So why focus on ALS as one of our primary leads? So ALS is a horrible disease with a significant unmet need. There's over 5,000 newly diagnosed cases per year and about 30,000 affected individuals. It's a very progressive disease once symptom onset starts. The average time of diagnosis is about 55, but it's a 3 to 5 year survival time from time of symptom onset to the time that you have to make decisions about going on lifelong supportive ventilation, or if you choose not to, typically you would die. There's 2 FDA-approved drugs in ALS, but neither one of them improve quality of life, and neither one of them extend survival, so a significant unmet need as far as clinical development goes. Inflammation plays a prominent role in neurodegeneration, not only ALS, but multiple sclerosis and Alzheimer's disease. The peripheral immune system, once it becomes activated, crosses the blood-brain barrier, T cells and activated monocytes, B cells, and they activate the innate immune system in the CNS, microglial and astrocytes, and cause local inflammation that results in neuron death. And we see this not only in ALS, but also in Alzheimer's disease. So by modulating this peripheral activation of your immune system can stop those cells from becoming pro-inflammatory, and they won't cross the blood-brain barrier and won't activate inflammation in the CNS. We did a lot of work in animal models of ALS demonstrating this concept, published in Nature back in 2010. I'll walk through this slide from left to right. The murine antibody that works in mice is called MR1. And that left-hand box plot or graph with CD68 positive cells, those are those pro-inflammatory macrophages that I mentioned. This is happening out in the muscle beds. In animal models of ALS at the time of muscle atrophy, when the nerves are getting sick and they pull back and they don't innervate your muscle, that's why you can't walk, and that's why ALS becomes progressive. Macrophages recognize those denervated nerves, and they literally go into your muscles and they eat them and chew them up so that they no longer can ever connect back to the muscle. When you block with MR1, you can see in that box plot, you knock down macrophage accumulation in the muscle by 50%. What that results in is that middle box plot. At the time of symptom onset in these mice around day 70, you can see in this vehicle control set of animals on the left that at around day 70 you have this massive loss of neuromuscular junctions, which is the connection of the nerves to the muscles to make the muscles move. When you block CD40 ligand signaling with MR1, you pretty much bring all those neuromuscular junctions back, which is why the animals don't have muscle atrophy, and it's why they're ambulatory, and it's why they live longer. What that translates to is that far right-hand box plot. It improves motor neuron survival in the central nervous system. We see an improvement in motor neuron survival, even though the antibody's too big to cross the blood-brain barrier. What it's doing is it's stopping pro-inflammatory differentiation in the periphery. It's stopping T cells, monocytes, macrophages from becoming activated. They don't cross the blood-brain barrier, and you don't see inflammation in the CNS. And we've also seen this in animal models of Alzheimer's disease, very similar mechanism. Our ALS phase 2 study enrolled its last patient, received its last dose about a month and a half ago. It was a 12-week open label study, 4 dose escalations, about 18 subjects per cohort. Primary endpoint, because this is our first multiple ascending dose study, was safety and tolerability. Clearly, because we dose escalated through the 4th dose, last patient, we haven't seen any safety problems. But we have 3 critical biomarker components to this study which will educate whether the drug is doing what we think it should do. The first one is target engagement. Because the CD40 ligand is expressed on the cell surface, we can monitor if it's blocking signaling by taking blood from people and seeing if we knock down markers like CXCL13. The second one is that critical component I've mentioned over and over again, that in autoimmunity like ALS, there's pro-inflammatory molecules that are in your blood that we can measure. And because we're blocking pro-inflammatory signaling, we should see a reduction in these markers, molecules like TNF-alpha, MCP1, Enraged, and others. So that's our second biomarker component, not only target engagement, but we should knock down pro-inflammatory signaling. The third set of markers, which is really interesting, but this is a fairly short study, it's only 12 weeks, would be have we flattened the curve of disease progression? ALSFRS is a scoring system that monitors disease progression. Have we slowed that down? There's also another marker called neurofilament light chain, which is a component of motor neurons and neurons in your central nervous system. When the neurons die, fragments of this molecule end up in your blood. Can we stabilize that and maybe decrease those levels, suggesting that we're improving neuron health? Again, it's only a 12-week study. We may not see those, But if we did, that would be very, very compelling data on top of our target engagement in pro-inflammatory markers. Moving over to kidney transplant, again, a significant unmet need, orphan disease, about 23,000 transplants are done per year. The current standard of care uses CNIs, calcineurin inhibitors, tacrolimus being the most popular. It works fairly effectively at preventing organ transplant rejection, but it has a significant amount of toxicities that show up within 6 months that I'll review. And ultimately, by 3 years, about 50% of transplants are actually rejecting because of the nephrotoxicity that's associated with the actual drug that's supposed to be saving the organ. About 15% of transplants that are actually done are retransplants because of calcineurin inhibitor neurotoxicity. So our goal here is to replace calcineurin inhibitors with our molecule, with our antibody, to avoid all of the toxicities that are associated with calcineurin inhibitors. This is an example of why that data is so compelling. This is a meta-analysis of over a dozen publications. There's over 100 monkeys in this graph. You can see that the red line, untreated animals, reject a kidney transplant within a couple of days if they receive no treatment. The organ isn't theirs. The immune system recognizes that it's foreign and completely destroys it within a few days. If you block anti-CD40, the receptor, you can see you get pretty good extension of survival as a monotherapy. This is just one drug. It's an antibody to CD40. You can extend organ survival out to about 100 days, but look at how more potent blocking the ligand is. It's about a 33% improvement over blocking the receptor for the reasons that I described previously. These are some of the toxicities that I mentioned that are associated with exposure to calcineurin inhibitors. You can see that about 30% of people within 6 months develop type 1 diabetes when they're on tacrolimus, and that's because it's toxic to the insulin-producing cells in your pancreas. It also causes renal issues, the actual organ that you're transplanting, in about 20% of people within 6 months. It also has neurological issues that cause tremor. This looks a lot like Parkinson's disease. It increases the risk of opportunistic infections, and unfortunately it actually causes hair loss. So there's a lot of long-term toxicities that are associated with exposure to either of these calcineurin inhibitors. And here's a graph of the long-term rejection rates if you're on these drugs. Yes, in the first year post-transplant, these drugs do quite well at preserving the organ, but you can see by 5 years, 50% of transplants are rejecting, and by 10, over 100% of organs are pretty much rejecting due to nephrotoxicity. So this is an open-label 52-week study. We're enrolling 6 to 12 subjects. We have country regulatory approval in Canada, the UK, and Australia. We have sites open and are actually screening already in Canada. UK is coming online this month, and we expect Australia to be up and running by the end of the month. Again, it's a primary safety tolerability study, but ultimately, as I showed you in a couple of slides ago, graft rejection happens quite acutely. So we'll be looking at graft survival in this study, and typically 90% of grafts fail in the first 3 months. So patient by patient, we will have a very good understanding whether this drug is effective at preventing transplant rejection in humans, much like we've seen in non-human primates. In islet cell transplant, we're really trying to unlock a new disease space. Islet cell transplant's not approved in the US for people with type 1 diabetes, and yet 1.3 million Americans suffer from the disease, and about 100,000 people have what's called the brittle form of the disease, where they really can't, they're unaware of their hypoglycemic unawareness events, which sets them up for a lot of comorbidities. And islet cell transplant has been shown to be quite an effective way to replace the cells in their body that were destroyed so that they can make their own insulin and really be much more aware and cognizant of their glycemic awareness. You can get them off exogenous insulin. The challenge has been it takes calcineurin inhibitors to prevent rejection. It's the same drug that we use for organ transplant, and the very cells that you're transplanting, if you remember, get killed by this drug. So we think if we can take calcineurin inhibitors out of the equation, we can successfully get people off exogenous insulin with an islet cell transplant for years and years, which has been demonstrated with the current state of the art. We've been doing studies in non-human primates with Norma Kenyon down here at the University of Miami for quite some time. She compared calcineurin inhibitors in her non-human primate islet cell transplant model to our tegoprubart anti-CD40 ligand antibody monotherapy, and you can see the animals have much better rejection-free survival. The islets themselves last significantly longer, meaning these animals don't require exogenous insulin compared to the animals on calcineurin inhibitors. They also don't have opportunistic infections. And the animals are ultimately significantly healthier. If you look at the box plots on the left, that's body weight. Animals on calcineurin inhibitors lose body mass because they're really sick and they don't eat. Animals that are on our drug feel much healthier, they grow much better, and they just perform much better. They're much more mobile in their cages. In addition to that, the graphs on the right show that we control glucose levels much better than calcineurin inhibitors do. That's because the islets are healthier. They're much more dynamic in their function. They sense when the animals have eaten, and they need to secrete more insulin to control their glucose levels. And finally, C-peptide, which is a surrogate of how much insulin these animals are making with the transplanted islets. You can see in the right-hand graphs, the animals on tegoprubart make much more insulin than animals on CNIs, and that's because they're healthier. This again is an open-label 52-week study enrolling up to 6 subjects. We have a site open up in Canada, in Edmonton. We're about to open up a site. We got regulatory approval here in the US several months ago. We're about to open up a site in Chicago. Again, the primary endpoint is safety tolerability, but ultimately we will know if these grafts survive within 30 to 90 days post-transplant. Can you get these animals off exogenous insulin or not? We can measure insulin levels in their blood because we've transplanted the islets and insulin will be in the blood, so it's a non-invasive procedure to see how healthy the islets are that we transplanted. Recently Vertex put beta cells into a person with type 1 diabetes. They didn't even get them off exogenous insulin, and that data made the New York Times with a single person. So quite an opportunity to see if we can transform the lives of people with type 1 diabetes with islet cell transplantation. And finally, our last indication in autoimmune nephritis is IgA nephropathy. It's the most common glomerular nephritis, which is an autoimmune disease that attacks the kidney. It's gradual and progressive. It occurs earlier than diseases like ALS, so typically people in their 20s and 30s will start to see proteinuria in their urine, and it increases over time. There's a clear regulatory path that you can measure proteinuria non-invasively in the urine, which makes it very easy to execute and facilitate clinical trials. Untreated, about half of the people with disease will eventually reach end-stage renal disease and either be on dialysis and end up on that kidney transplant list that I described, which is very long and very limited. Most people end up on that list and they actually never get their transplant and actually succumb to the disease if they can't tolerate the dialysis. The pathophysiology of how IgA nephropathy happens is really well understood. It's a 4-step mechanism where genetics ruin an enzyme that puts sugar molecules on an immunoglobulin called IgA. So that's the first step. Enzyme deficiency makes an improper protein. The second step is the immune system, of course, does its job. It recognizes that that IgA is not normal, it's not you, and B cells start making antibodies to eliminate it. And when B cells make antibodies to eliminate something, the antibodies glom onto it and they form something called immune complexes, which is aggregated molecules that are in your blood and they get deposited all over your body. So that's step number 2. Because those immune complexes are in circulation, they end up in the kidney. Your kidney is a filter with very, very small filtration capabilities. These immune complexes are too big and they get clogged in the kidney and they cause local fibrosis and kidney damage, and that's why you get proteinuria in your urine. And once this damage starts, if it's not treated, it just progresses to decades until you have end-stage renal failure. So deposition in the kidney is your last step, step 4. Blocking Cd40 ligand hits all of these processes and should fix all of them except for the genetics upstream. It'll stop step 2, B cells making antibodies, because that's how the molecule works. If you block Cd40 ligand signaling, you block B cell maturation and they won't make antibodies. They won't recognize that for an IgA molecule. That'll reduce the immune complexes that are circulating in your circulation because you're not making the antibodies that's going to glob onto it. The other thing that it does, though, that blocking Cd40 ligand will do that's very unique and is unique to any other molecule that's in clinical development for IgA nephropathy is, even though you show up in the clinic and you've already got some kidney damage, what happens there is the immune system goes into the kidney recognizing that damage, and it causes subsequent inflammation that damages the kidney more. Blocking Cd40 ligand decreases that pro-inflammatory signaling that I described and reduces the immune infiltrate that shows up in the kidney so that the kidney can actually repair itself. So by blocking CD40 ligand, we should hit all of the processes that are downstream of the enzymes that inefficiently don't put the right sugars on the molecule. Blocking this in animal models, by blocking CD40 ligand, pretty much cures mice of the disease. So there's animal models of glomerular sclerosis, which is what I'm showing you here. The far left-hand graph is called the Kaplan-Meier survival plot. You can see untreated animals that are treated with a control IgE antibody. The animals start to die within about 7 months. You can see animals with the blocking antibody to CD40 ligand, the same antibody that was used in those ALS mice. The animals live forever. They never show any kidney dysfunction that's severe enough to kill them. The middle box plots there is proteinuria levels in the urine, which is what we're going to measure in the clinic. The untreated animals are the ones that are the little dots that go all the way up to the top with very high levels of proteinuria in the urine. You can see the individual animals treated with the CD40 ligand molecule have significant reduction of proteinuria in their urine because you stopped the fibrotic process that further damages the kidney. And what that translates to is that far right-hand bar graph where I show you that macrophages, which are the immune cells that try to go into the kidney to repair the damage, you can see that you significantly reduce that macrophage infiltration into the kidney itself. So this is a trial that's a 96-week, again, open-label study with 2 dose cohorts. Primary endpoint, though, is at 24 weeks. It's reduction in proteinuria levels. So 20 subjects are going to be at the higher dose. If that data looks positive, we're actually going to dose de-escalate down to a lower dose. We're looking at patients that show up in the clinic with a proteinuria of about 0.75 grams, which is fairly high. And we anticipate that we'll be able to quickly monitor whether we're fixing that fibrotic process and reducing that immune cell infiltrate to reduce that daily proteinuria output. Again, because this is our first study in IgA nephropathy, primary endpoint will be safety tolerability, but ultimately the primary endpoint for the study is changes in proteinuria at week 24. We're going out to 96 weeks because the long-term regulatory path requires that you look at a different measure of kidney function called eGFR. We have this study approved in multiple countries at this point, including Malaysia, New Zealand, Australia, other sites in Europe are coming online now, and Southeast Asia should be next month, and anticipate that we'll have subjects in this study by the end of the year. And again, we should be able to measure proteinuria on a monthly basis to show that we're reducing it and improving kidney function. So we have a bunch of priorities that we're trying to execute, as you can hear. We have top-line data coming out for our ALS study at the end of this quarter. We should initiate both of our transplant studies as well as our IgA nephropathy study at multiple sites around the globe, and should be able to dynamically monitor whether the drug is efficacious in preventing transplant rejection and improving kidney function in people with IgA nephropathy. And thank you for your time. I think I have a minute or two for questions if there's any. I mean, in ALS, any positive clinical data in ALS is a huge win, right? There's no effective treatment for ALS. We enrolled that phase 2 study that I mentioned with 54 people during the height of COVID, and at one point somebody tweeted when they were the last person, I think in cohort 2, they tweeted, "I just got the last spot in Eledon's ALS clinical trial. I'm not afraid of COVID-19, I'm afraid of ALS." I mean, ALS is a horrible progressive disease. Once you get a diagnosis, there's no slowing it down. And I've seen people with the disease lose their battle in 6 months of symptom onset. You know, not everybody is Stephen Hawking that goes out 20 years. The average person is 3 to 5 years. So positive biomarker data showing target engagement and knocking down pro-inflammatory markers, that's never been demonstrated in an ALS clinical trial. That would be a big win for us. The ALS trial will have top-line data that we'll be releasing in June. So right around the corner, 6 weeks or so.
With this being so important to ALS patients, and in addition to your other programs, how do you feel that Big Pharma is looking at Eledon at the moment?
Well, I mean, I think any of our indications is a huge win, right? They're all significant unmet needs. We could change the way transplant patients are cared for, and that hasn't happened in 25 years. The current state of the art, the development of calcineurin inhibitors, really made transplant rejection feasible, preventing it 25 years ago. There's been no improvement in the technology since then, so getting rid of that will be absolutely amazing. Same thing with IgA nephropathy, so I think pharma will look at us as a win for anything.